And its particular restriction site, if I remember correctly, is GAA. Like that. So only when it finds this site in the sequence will this BAM-H1 enzyme cut the DNA at that site. And there's another one called KIND3. It has a completely different sequence. It looks for, I think, AAG, CTT, BTC, GAA, like that. And only when it encounters that particular sequence in the DNA does it cut the DNA there at that particular sequence. And there's one called EcoR1, and there is EcoR1 enzyme's restriction site, that particular site right there. Now, the presence of these particular restriction sites is essentially random in the DNA. Any given piece of DNA has essentially a random sequence of G's and A's and T's and C's. And so how often you find the restriction sites is more or less a random distribution. You might find 2 EcoR1 sites just only a few hundred base pairs apart, or you might find an EcoR1 site and the next EcoR1 site might be 10,000 base pairs further along the chromosome. It's more or less random where you find these sites. But when a restriction enzyme does find its site, I think this one, if I remember correctly, has an EcoR1 site in it. When a restriction enzyme does find its site, it does cut the DNA at that site. So let's see, the EcoR1 restriction site is GAATTC. So if it was scanning along, oh, here it is right there. And what the enzyme does is typically make what's called a staggered cut. It cuts that strand there, and then cuts that strand there. So now you get one piece of DNA like I've drawn there, and the other piece of DNA would be like that. They show there, yeah, that's what they show there. Good, okay. So notice that we went from having one piece of DNA to 2 pieces of DNA because the EcoR1 restriction enzyme cut there. And so in theory, these 2 pieces of DNA, well, it's not in theory, that they're not one piece of DNA anymore, so that they could drift away from each other. Maybe I'll show that here. This one might just, since it's no longer really attached, like that. Like so. Even though when you cut with a restriction enzyme, you get 2 different pieces, they're going to have a certain attraction towards each other, and here's why. Because typically the cut leaves what they call these sticky ends. A sticky end means where there's a short single-stranded piece sticking out here and here, and these sequences are complementary. And notice that they are. AATT is complementary to TTAA. So the backbone has been broken, but complementary sequences have a certain attraction for each other. Who remembers what type of bonds hold complementary sequences together? Hydrogen bonds, that's right. And hydrogen bonds are not super strong, but they are a type of bond, a weak type of bond. And so there's still going to be some hydrogen bonding between these sticky-ended pieces. So especially at colder temperatures, they're going to want to still stick together a little bit. But again, let me emphasize it's no longer joined by the phosphate-ribose backbone, and so they're not really welded together anymore. If you have two pieces of DNA, and you do want to join them together permanently. In other words, if you do want to have the backbone, the phosphate ribose backbone, joined together, then that's where this other enzyme comes in, DNA ligase. DNA ligase can take two pieces of DNA and connect their phosphate ribose backbones. Again, to ligate means to join together. And so if the two pieces of DNA have sticky ends, the ligase works much more efficiently because these two pieces already want to come together by their complementary hydrogen bonding. And so it helps the ligase along. But even if the pieces don't have sticky ends, if they have what's called blunt ends, just cut squarely at the restriction site, then ligase will still work. It's just not as efficient a process. So let me put this back. If we allow these two to come together, to physically come near each other again, like that, and then we add DNA ligase, it's going to reconnect the phosphate ribose backbones, and you get one piece of DNA again. And so this is the typical approach to a gene cloning experiment. You cut your gene of interest and the plasmid with the same restriction enzyme, so they have the same sticky ends there, and there, and so their natural tendency is for this wants to click into place there. But then you have to add the ligase enzyme, and it joins the phosphate ribose backbone, and now you truly have a piece of recombinant DNA for your gene cloning experiment. There you go. All right, and there you see that in gray is the piece of DNA from the other, another species, but you've cut it with the same restriction enzyme, and you put them together, and typically you cool the temperature a little bit, so that allows them to anneal, to join to each other by complementary base pairing because they have these sticky ends, but they're still not ligated yet. And then you add the ligase enzyme, and it joins their backbones together, and now you truly have a piece of recombinant DNA. All right, well, when you're interested in doing a gene cloning experiment, usually there's one particular gene that you're interested in. If you were studying this jellyfish and you're interested in the gene, in the green fluorescent protein, that's just one gene amongst the tens of thousands that are part of the genome of the jellyfish. And so, typically, what you want to do is make a collection of the DNA pieces from the organism you're interested in, and that's called making a DNA library. You don't know which piece has your gene of interest yet, but you use restriction enzymes to cut the DNA up into thousands of pieces, and you know one of those has your gene of interest, and that is making a DNA library, collection of DNA pieces that contain some or all of the genes of the organism, and one of those pieces has your gene of interest. You just don't know which piece yet. And there are two types of DNA libraries. One's called a genomic library, and one's called a cDNA library. And the distinction is the genomic library comes from the genome itself. You're cutting up the actual chromosomes, all the chromosomes of the organism you're studying. Whereas the cDNA library is a smaller library. It only represents DNA of the genes that are expressed, because an organism doesn't necessarily express all of its genes. So let's look at this one first. This one's closer to the experiment that we're doing here in the laboratory. As a matter of fact, that's why you guys isolated genomic DNA as an exercise on Monday, because if we were pretending that that was genomic DNA from the jellyfish that we're studying, and so we need that as a starting material. All right. Here we go. All right. So, if you are a researcher and you want to study a gene of interest from the organism that you're studying, the first thing you do is purify genomic DNA. That's what we did on Monday. You break open the cells, and remember you guys had to get rid of the lipids, and use the SDS and the EDTA, and got rid of the carbohydrates and the proteins, like the sodium chloride removed the proteins, and then you used the alcohol to precipitate the DNA, but not the proteins. You purify your DNA, and then you take the purified DNA and digest it with a restriction enzyme like E4R1. So that cuts all those chromosomes of the entire genome of the organism into little bits and gives them sticky ends. And one of those bits would have your gene of interest. Now, here in this diagram, they're only showing 3 bits in this genomic library, but chromosomes are huge, and so you would probably end up with, I don't know, millions of DNA fragments in a real genomic library. And then you take your fragments of genomic DNA and you have to mix them with what's called a cloning vector. And a cloning vector is a thing like a plasmid that can be used to make large amounts of your gene of interest. I sometimes think of cloning vectors like the plasmids as the copy machines for your DNA, the DNA in your library. A piece of DNA that can be used to make multiple copies of your gene of interest. And so what you have to, just like if you have a document, you have to put it into the copy machine to get it copied. You have to ligate your bits of DNA from the genomic library into your cloning vector to allow it to make the copies of your gene of interest. The plasmid is probably the most popular type of cloning vector. There's another type called a bacteriophage. And I'll explain how they use that as a cloning vector in a moment. But in any event, to get on with the genomic cloning, you cut open the plasmids with the same restriction enzyme. And so they have the same sticky ends as your genomic DNA fragments. So these are going to naturally want to click into place by complementary base pairing. But you still have to add the ligase enzyme to join them together. And so now you have a bunch of plasmids, all of which are recombinant now because they have bits of your jellyfish DNA in there. And to be honest, that's actually the definition of the genomic of a DNA library. A DNA library technically is defined as the collection of DNA pieces from the species you're studying, but with all of those pieces joined to a cloning vector, like you see there. Yes.
So when you're using the restriction enzyme, is that one type of enzyme, like say for instance per test tube, or you have a bunch of the different kinds of restriction enzymes mixed together?
You typically use only one restriction enzyme per test tube, although occasionally in some experiments you might use 2 per in the same test tube. All right, so now it's time to start using the bacteria as copy machines. Remember, there's this process called transformation where bacteria can take in naked foreign DNA, like the plasmids in your genomic library. You then transform these bacteria with your plasmids, and if there's one thing bacteria can do, it's reproduce themselves. Remember, by this binary fission type of division, up to 20, every 20 minutes they can double their number. And so these bacteria take in the plasmids, and you essentially just give the bacteria growth medium, which means the nutrients they need to grow, and put them at a warm temperature. And so each bacteria, transformed bacteria, will divide and divide and divide. And every time the bacteria divides, it not only reproduces its own chromosome and proteins and membranes, it also reproduces any plasmids that are inside itself. And so you've gone from just one copy of your gene of interest to millions of copies of your gene of interest. And yeah, you're also reproducing the copies of the other genes that you're not interested in studying as well, but the point is that you are cloning the gene. You are getting millions of copies of your gene of interest. The next step you're going to do is called screening the library, which means actually looking through all these millions of copies of bacteria with the different plasmids to find the one that has your gene of interest. But that's going to be something we're going to talk about in a different lecture. Any questions on the genomic libraries, what they are and the basics of how they're made? Victor?
Bacteria usually have their own plasmids naturally?
They can, naturally occurring bacteria can, but they don't necessarily have them. So some bacteria just don't have any plasmids. And when people are doing DNA cloning experiments, they deliberately select bacteria that don't have their own plasmids, so that the bacteria essentially have to take in only the recombinant plasmids that you give them. Good question. Oh yeah, so I mentioned that plasmids are